This is the part where I tell you about the repeating DNA. This is one of my favorite parts because it's so wacky. Let's look at tandemly repeated DNA and interspersed repeated DNA, which are the two largest types of repeated DNA. But before that, why is DNA repeated? Mostly to benefit us. DNA is repeated so we can create a structure where we, like telomerase or centromeres, we have a structural purpose for the DNA that we're not going to read, or we need to just add some junk in between two genes to physically separate them, which is not very common, honestly, because the physical position of the genes don't really matter that much on how much they're replicated or how much they're read or used. However, there are some useful repeated DNAs like telomeres and centromeres. But there is also the interspersed repeated DNA, which is mostly transposons, which are just hell. Now let's get into that. Tandemly repeated DNA is DNA that's repeated in a line. So one repeat next to the other repeat next to the other repeat in a huge line. A good example is the telomere ends, which repeat what was that? TTAGG 4,000 times. They make up 10 to 15% of a typical mammalian genome, and they're small. And by small, I mean 1 to 2,000 base pairs, which is basically nothing. There is also a smaller version of them, which is simple sequence repeats, or satellite DNA as we call them, which are tandem repeats that are less than 10 base pairs. So it's like 80, 80, 80, and like a million of that. They're usually harmless. Interspersed repeated DNA, however, is large. And the repeats are not in a line, but they're stuck to random places in the genome. It can be a different chromosome, it can be the other end of the chromosome, you don't know. There's no rule to it. The repeats are scattered, and the way that they're scattered is also important because in tandemly repeated DNA, the DNA doesn't repeat itself. Something else comes and writes that DNA for some reason, like telomerase enzyme adding to the telomere ends. But in interspersed repeated DNA, the replication of the DNA, the reading of the DNA, is immediately causing the repeating of this DNA. Let's look at transposons for that. Transposable elements, or transposons, are most of the interspersed repeated DNA in the mammalian genome, and they can move around the genome, leaving copies of themselves by a mechanism that I can describe as a railroad, where they create a RNA copy of themselves. The RNA creates a protein that will read the RNA and create a DNA copy of it. Then another protein that they code for will take the DNA and shove it in a genome. However, there is also two types of transposons, lines and signs. Lines are creating these proteins that read them and convert them to DNA. They're creating these proteins or signals that makes the DNA copy of them just stick to another part of the genome. Meanwhile, signs don't care. Signs are so small that they can't code for these proteins. They just make the existing proteins in the cytoplasm, which are usually created by the lines, to do their bidding for them. So signs don't code any element that's going to replicate them and put them somewhere else. They just call upon the existing element to replicate them and put them in the genome. An important part of signs are Aloe sequences. Aloe sequences is the most common sign in humans, making 10% of the human genome. Imagine that. One repeated sequence, smaller than 50 base pairs, repeated so much that it makes 10% of human genome. Now let's look at the human genome. 1.5% of it is exons. Exons are the parts we use. Exons are the parts that are converted into proteins. 24% are introns. Introns and exons together are converted into RNA. However, introns are taken out of the RNA and only exons are converted into proteins. or in some cases they serve as RNA as well. Why do we have introns? I'm going to get into that later on. Now let's look at the junk, because this part is useful or partially useful, let's say, but these, the only purpose they can serve is structural purpose, and some of them don't even serve that. The unique non-coding DNA is non-repeated non-coding DNA, which makes up 15% of the genome. Then there's tandemly repeated DNA, which I've talked about here, which makes 15% of the genome as well. And then 44% of the genome is interspersed repeated DNA, basically just transposons, those parasitic whatevers. I mean, imagine that. You're a DNA piece and you're just being parasitically... That's just so disgusting. It's so interesting, but it's also so disgusting at the same time. And why do we have these transposons? I'm going to give you one example. Sometimes a virus infects a person, and then sometimes the infected person will carry this gene within their cells, because the virus will inject the DNA they have into your DNA, so whenever you read it, you're going to create another virus or another viral protein or something the virus needs. Now, this is all nice, all good, and then you die because of a viral disease, but sometimes you don't die, which is bad in an evolutionary sense, because you keep that DNA within your genome and sometimes even in your gametes, which are your sperms or eggs, depending on your gender, depending on your sex. However, these elements that stay in your DNA, in your gametes, can pass on to your daughter cells, pass on to your children, and then they pass it on to their daughter cells, and they pass it on to their children, and thousands of years of this will create an accumulation of these interspersed repeated DNA sequences, these transposons within your genome that might have had a viral origin. Now, how weird is that, right? Okay, this is another subject, so I'm just going to cut this video here. We mentioned the DNA structure, DNA packaging, euchromatin, heterochromatin, and repeats in DNA in this 4 or 5 videos, I think. Have a good time.